In the hurricane example, we've given you an opportunity to see some of the advantages of analyzing whole class data. Now we want to see a variety of different activities and give you the tools to actually replicate ours. In the future activities, you'll be generating your own from scratch, but for right now, we're going to work with some of the ones we've developed over here. So we're going to start with the biology one, and if you click over on the biology tab here, you'll see a number of different activities. The one we're going to choose right here is on mitosis. Well, that's a typical topic that you're going to see in high school or even middle school biology classes. Mitosis is the process of cell division. And typically the way that this lab is done is that students are given prepared slides of either onion root tip or whitefish blastula, and they're asked to find cells in each stage of mitosis, in prophase, metaphase, anaphase, and telophase, and so forth. In this activity, we're going to do more of an investigation to try to see something about the nature of the cell cycle. So rather than just trying to draw pictures of various different stages of mitosis, we're going to try to come up right here with some kind of hypothesis based upon data from the entire class. So we've introduced basically a diagram here that shows first you have interphase. 1 and 2 is an interphase where there's no clear chromosomes visible and so forth. And when you get to prophase, the first stage of mitosis, you can see the condensation of the chromosomes, and you can see them here in 3, 4, and 5. Finally, when you get to metaphase, you'll see meta meaning middle, that those chromosomes are lined up in the middle of the cell. If this is the cell boundary right here, then that's the middle or the meta zone there in which they line up. And ana meaning apart, they're starting to move apart right here. This is the first part of anaphase. And then as you go farther on, those chromosomes separate more and more, and until you end up with what's called telophase, where the chromosomes are very far apart. And eventually, in number 10, you're going from late telophase to what we call cytokinesis, cyto meaning cell, and the splitting of the cell. And you can see now that there's actually 2 cells that are ended up from the one. So again, rather than having the students draw these various different stages of mitosis or attempt to draw them in the low view that they would probably see them in low magnification, that they'd see them in a regular high school microscope. Instead, we're going to actually provide them with the tissue online. Now, again, this could be done with a regular microscope slide, but we're just going to do it here because we can do this over the internet. So here's tissue A, and then we can see tissue B. Again, these are all onion root tips, but they appear different because of different staining techniques, also different lighting, different microscopes, different optics, basically different researchers. But this is a kind of variety that really introduces students to the dynamic aspect of scientific research, that not everything's going to look the same, and not everybody's going to look at it the same way. And yet we can start to look for similar features. So, for example, this is a very obvious late anaphase or early telophase stage right there. And students will start to learn to identify that in a variety of different perspectives. We can see another example right there, and so forth as we go through the various samples that they're given. Here's another one over here, and so forth. So in this activity, each student group would be given a different pre-prepared microscope view. And they're going to be asked to look at this view and say, okay, which cells appear to be in which phases of mitosis? Now, these things all can be enlarged and so forth. Simply by clicking on them, you can see greater detail. And so in a traditional activity, the students would record their own data, but they wouldn't have the opportunity to see the data of their colleagues, their peers. But in a CSCS activity focusing on pooled data, there will always be a way for them to submit their data. In this case, we're doing it using a form. So if they would click at the bottom of their page. They would click right here. They would say enter that. And let's say that this was me recording this data. And then let's say that I counted 85 in interphase, maybe 23 in prophase, and 12 in metaphase, and let's say another 24 in anaphase, and let's say 4 in telophase. So once they submit that data, theirs is going to go into a database where other people can access their information. So let's go back up here and see where that is going to be. So in the activity itself, you will see there is a table right here which includes everybody's information. So we can see Gladys and Debbie and James and Cordell and so forth. And so here's all of their data. And this is a spreadsheet which is associated with the form. So rather than just having their own data to look at, now they're going to see that collective data for the entire class. So as they scroll through here, you'll see that the most recent entry right here is by Norm. Now, we've set this up so that it'll graph automatically the number of cells in each of the various stages of mitosis. So if we're looking at this, we can see that of the class sample here, that interphase is the most common, and then prophase, and then you can see that metaphase, anaphase, and telophase are, there's fewer cells in those stages. Prophase through telophase is mitosis. Interphase is the phase between those. Now, if we look over here and actually try to graph it in a pie chart, we can see that, again, in a kind of more graphic form here, you can see that interphase takes up more than half of the total cells that were counted, but there's still a substantial number of prophase, metaphase, anaphase, and telophase. Now, the question is, what do you do with this data? Again, rather than just drawing nice little pictures and so forth of the various different stages of mitosis, we want the students to do something more analytical, something more in line with the goals of the next generation of science standards, Dimension 1, in terms of the science and engineering practices. So we're going to have the students enter their hypothesis here. So you'll see that there's another form right here, and they may generate some kind of hypothesis to the effect that they think that it appears as though most of the time the cell is in interphase. Now again, that is based upon just the observation that most of the cells that they found were in interphase. Over half of them were in interphase. And that may or may not be a correct hypothesis, but it gets them thinking about things like the cell cycle here in terms of how cells actually divide, reproduce, and how tissues grow. So if we were to look at actual diagrams of mitosis, so here is a diagram of mitosis. This is actually from animal cells, but the pattern is very similar. And it's fairly complex, and students can memorize these various different stages, and there is definitely value in that. But we think it's more important that they actually be able to analyze this, in other words, given the cell cycle stages and so forth, to try to figure out what does that mean. So here they would have noticed, okay, from their data, that the vast majority of, or I should say the largest percentage of the time, the cells were in interphase. In this diagram here, which is showing the cell cycle, this broad orange circle here represents interphase, and that small m there represents mitosis, which would be the 4 stages you saw, the prophase, metaphase, anaphase, and telophase. Well, obviously, in this case here, this is a more typical cell than the ones we were using. We were choosing ones that were at the root tip because that's an actively dividing, actively growing area, and so a vast amount more mitotic activity. And so in our cell cycle, if we'd had the students draw our cell cycle, we would have had this mitosis taking up about 40% or so of the cycle and the rest being devoted to interphase. But they're getting the concept from the data there that this cell does actually go through a cycle, and it goes through division, and that by looking at snapshots in time, we can make some kind of inferences about a dynamic process that's very difficult, if not impossible, to observe, at least accurately, real time. So now it's your turn. We would like you to select one of these microscope views, and then once you've done so, is to count the number of cells that you see in interphase, prophase, metaphase, anaphase, and telophase. And when you're done, enter your data into the table and hit the submit button. And then once you've submitted that, you can then go back to the activity and you can see your data in the context of whole class data. So you'll look down here, you'll see your name appearing here at the bottom, indicates that it's actually entered. You can then check to see how that has actually changed the overall ranking and so forth of the data as an entire group. And then you can see if that's made any change to the percentage of the cells we see in each phase of mitosis. Finally, when you look at that, you can go ahead and enter your own hypothesis and so forth here. Maybe it's going to be different than the ones that we suggested here. And that would be something that you could then discuss. Now it's your turn to do the investigation. So click on the mitosis activity, analyze the tissue, and enter your results and hypothesis.